Hello everyone, just while people are coming into the room, I'm going to give it a couple of minutes before we get started, so hang in there and we'll get started in a sec. Just give it a minute or two more, the numbers are still going up. Okay, I'll start the intro and then people can continue to join as and when. So hello everyone, and on behalf of the Icon Book and Paper group, I just wanted to say welcome and thank you all for joining us today. I'm going to do some housekeeping as usual, so regular viewers, I apologize, but just hang in there. We're using the usual hashtag, which is conservation together at home, so please do feel free to use that to join in the discussion on your social media channels if that's what you're into. As usual, please bear with us, as I'm sure you're all aware, we're using our home internet at the moment, so if anything does happen or the webinar ends suddenly, you can use the same link that you used to get in to rejoin the webinar once it's open again, but I'm sure we won't need that. This webinar will be recorded and the recording will be made available as soon as possible, probably in the next couple of days. I'm just going to draw your attention to the Q&A function, which you should see at the bottom of your screen, which you can use to submit questions throughout the talk, and we'll have 10 or 15 minutes to go through those at the end. There's also, I've enabled the chat, so you can use that to chat and to write comments or anything as we go. So without any more delay, I'm going to introduce our speaker today. Tiffany runs TEM Conservation in Ottawa, Canada. She completed projects for institutions including the Supreme Court of Canada Library, Ottawa City Archives, and private clients in the National Capital Region. Previously, she was a fellow at the Canadian Conservation Institute and has worked at York Explore Library and Archives in North Yorkshire and the House of Lords Parliamentary Archives in London, which are both in the UK. She graduated from Westdean College with an MA in Conservation of Books and Library Materials in 2015. So without any further ado, I'm just going to unmute you, Tiff, and you can go for it.
Great, thank you very much. You can hear me? Okay, great. Thank you for joining this presentation, which is about mold and mold swabbing. So just two little notes before we start the presentation. Even though this is about mold, as a caveat, I'm not a biologist and I'm especially not a microbiologist, just a conservator who's interested. And so I'll be approaching this talk from the conservation perspective of someone who has experience with moldy items in both single artifacts and in larger collections remediation. The second is a tiny confession that I've lifted this particular slide directly from a presentation that was created by CCI paper conservator Crystal Maitland for a presentation we did at Queen's University. And I wanted to note this because much of the preparations and discussions and editing through the research was done with Crystal. So I'll be mentioning her name a fair few times over the course of the slides. So to give a bit of background, this research was completed while I was a fellow at the Canadian Conservation Institute, also known as the CCI. I was really lucky to be able to carry out this research as I have a standing interest in mold, and when I arrived at the CCI, there was an interest in learning more about the piece of technology they'd recently acquired, a rapid adenosine bioluminescent swab tester, which is the topic of this presentation. So just to say, I feel lucky to have been at the right time, right people, right place. In terms of mold, detecting and remediating mold to a safe level has always been a concern for conservators. There's so many approaches to remediation, and there's a growing body of research on the health hazards posed by fungi, which means we're always concerned with determining the level of risk that a mold outbreak poses, or how best to decide if the treatment methods are successful for conservation. So recently, rapid adenosine bioluminescent swab testers, or sometimes they're known as ATP testers, have begun appearing in conservation with two primary applications. So one is identifying if mold is present on an artifact, and two, if, or using it to quantify if a remediated object is clean enough to be returned to regular use or storage. So there's been a few prior papers on these swab testers. CCI learned about the swab testers at an IATA conference in 2015, where it was presented by Christina Meyer-Wolf. It's also been written about in several instances in the UK by the National Trust, in Poland by at the University of Warsaw Library, and in a poster and a paper from the United States. They're often also used as proof from salvage and restoration companies as a proof of remediation. And in Canada, it started with the CCI, I believe, and it's been used by several other institutions locally in pilot tests. So I thought I'd give a little roadmap of where we're going. I'll talk about the technology itself. How do you use it? How does it work? What we're attempting to find out? And then talk about the things that we learned through the literature review and the experiments. And then from there, I'll describe a framework that Crystal and I came up with on how to think about mold remediation and conservation. And then I'll end with just some questions that came up very frequently when we were talking to other conservators about mold. So to start, what are these devices? I'll give a little background info on using them and then a little bit more specificity in the next few slides. So rapid swab testers or rapid adenosine bioluminescent swab testers are designed for food processing and the hygiene sector, and they're used to test the biological load of a surface. So as an example from the food processing industry, the devices and swabs are used together with a preventative food safety approach, something like HACCP, to make sure that the daily cleaning of food processing machines has been adequate. So meaning that there's minimal organic residues remaining on the surface. The devices consist of a handheld luminometer, which detects very low levels of light emissions, and a disposable test tube kit, sometimes branded as swab pens, with a sterile swab and reagent separated by foil barriers. This is a Kikomin one, for example. The swabs are taken from the sterile pen, they're wetted, they're swabbed over the surface, and then put back into the test tube, which breaks the foil seals. Then they're shaken, which mixes it with the water and the reagent, and then they're placed back in the luminometer. The reagents cause any organic matter that's on the swab tip to react and create a very low level of light. And then the luminometer reads this light, and it reads it in a metric called relative light units, or RLUs, so I'll be using that term a lot. The manufacturers provide a range of RLU numbers below which are considered pass or clean, and then slightly above which are considered a caution level, and then a slightly higher number which is considered something like fail and re-clean, please. The most common number that came up in the leaflets and that was heard anecdotally for use in conservation was that anything below 500 RLUs was considered clean, because something like a cutting board with a regular surface is considered clean at a similar benchmark. So these swab testers are really attractive to use because the test itself is quite fast and they're relatively inexpensive for an institution. So it takes about 30 seconds to a minute to complete the test, and it doesn't require very much training to use. In terms of the cost, at the time of the experiment in 2018, it was about 2,000 Canadian dollars for the device and about $4 per swab pen, so potentially cheaper in other parts of the world as well. As I mentioned, the experiments in these project for this project, we used a Kikomin PD30 device, but there's many different types with different pen configurations, which you can see here. Okay, so just delving a tiny bit deeper into how these luminometer and swab pens work. They function by detecting adenosine triphosphate, or ATP, which is sometimes known as the universal energy molecule, as shown in the image above. And ATP is involved in the metabolic processes of all living organisms, so it's found in food, saliva, finger oils, and all microorganisms, including fungi. So it's a good signpost for indicating organic matter as being here. Some devices and swab pen combinations are also capable of testing for the degradation products of ATP, which are ADP, so adenosine diphosphate, and AMP, which is adenosine monophosphate. So these are all adenosines. And there are specific swab testers that can pick up ATP only, or ATP and AMP, or all three adenosines, and that's done by adding extra reagents to the pens. Okay, onto the swabs themselves and the reaction. So the reagent at the bottom of the pen is mainly luciferin, which is the same compound that's found in firefly butts, and it's what makes them glow. The swab picks up the organic material which contains ATP, where it's mixed with the water and luciferin alongside luciferase and magnesium. And so this is what causes the light reaction. And then the luminometer reads the intensity of the light reaction and gives results, as I mentioned, in RLUs, or relative light units. And then as a final detail, because of the enzymes present in the reagent, swab pens are stored in the refrigerator and they must come up to room temperature before testing, and they have a use-by date of a couple of years. Okay, so that was looking into the background of the device. It quickly became apparent that there were major adaptations when we were using the swab testers for conservation, which might affect their reliability or performance. Most of the fragile substrates, particularly for book and paper collections, can't be swabbed with a wet swab, so they're swabbed dry. Often there isn't large swabbable areas, so manufacturers recommend between 5 and 10 square centimeters. And finally, the tests were not meant to be used as mold-specific devices. Even though mold is generally the most likely cause of biological residues in collections, especially library and archive collections, other organic residues such as skin cells and dust will also be picked up by the devices. So learning that little bit of background information led to so many questions. And not all of them are directly related to the swab tester, but sort of associated queries on how we decide what's safe enough or what's clean enough when we're dealing with mold remediation. So in looking at all these questions that we had, they could largely be grouped into 3 categories. So the first is, how well do the devices work? How reliable are the results in general? So looking at the limits of the technology and sampling techniques. Secondly is probing how adaptations to the device for conservation might change what we consider safe. Benchmark, so is 500 actually safe, and what standard protocols do we need to use for conservation? And then finally asking what exists to currently tell us how much mold is too much mold. So getting updated on mold remediation and health guidelines from other sectors. And this, in that last one, it encompasses using disinfectants for mold remediation. So looking at common treatments and how necessary they are, or how they affect swab readers. So one of the first things that became apparent in the literature was that terminology emerging from different locations, languages, sectors, and differing levels of subject specialism meant that there was so many variations on the same term. So through working with the project, we used and have a preference for generic terminology in cases where specific terms are not clear. And this is just to keep in consistent and accurate way to talk about fungal issues in the conservation context. So I thought I'd just show a few. So describing what it is, starting with fungi and mold. So the term fungi is the name for the kingdom or the overarching branch of organisms, which is equivalent to plants and animals. And it includes all microorganisms that cause fungal problems in the heritage collections. So all molds are fungi, but not all fungi are molds. Mold itself is a name that commonly refers to fungi which grow hyphae and feed on dead organic matter, so it's the types we often find in collections. The other one that came up very often was about spores versus conidia. So spores are microscopic propagules designed for dispersal or survival, and these spores may be sexual or asexual, whereas conidia are a specific type of asexual spore from a specific family or subphylum. So all conidia are spores, but not all spores are conidia. Even though the types of fungi that grow conidial structures are really common in library and archive spaces, it seemed more appropriate to use the term spores when the fungi is not being identified at any species or level. In the literature, there was also many terms used to describe the activity and vital status of fungi, especially when talking about active, inactive, deactivated, and dormant. So just drawing from several health texts, I use these definitions through the project. The one I wanted to point out in particular is the term dormant spore, because I think it's often used to mean inactive mold. But since dormancy can be quite a specific term, perhaps it's not necessary to use, as most mold damaged collections will have a mix of viable, non-viable, inactive, and dormant fungal particulates and spores. And then another nice option that was suggested by a mold remediation textbook to talk about spores and particulates that are found in the air or that are just lightly settled and could be recirculated is the term transitory spores and particulates. And then lastly, for terminology, just describing remediation treatment. So a fungicidal treatment is one that's fully lethal to fungi when used as prescribed. And then a fungistatic treatment is one that inhibits fungal growth in some ways. Okay, moving away from terms and onto the technology itself. So much of the detailed information about rapid swab testers came from the hospital sector, where the main concerns are about relying on this technology is that even when tiny amounts of infectious or pathogenic microorganisms are there, they may be detrimental to human health. So reading those papers helped come up with key points to create a swab protocol in practice for conservation. So overall, the information shows that while rapid adenosine swab tests are a generally reliable tool, they do sacrifice some accuracy and precision for the ease of use. So on the accuracy and precision scale, it might look a bit like the low precision, high accuracy target on the bottom right, but a little bit more like this, where they have, in general, a medium accuracy and some precision. So this assessment comes from the literature, where lab studies show that RLU values generally have good correspondence with microbial contamination, but again, that's in lab studies. And in practical scenarios, there's less correlation. And this is likely because there's more interference from multiple ATP sources, multiple contaminants, including disinfectants. And fungal ATP has been shown to be more difficult to extract. Studies also show that the swab testers have repeatability issues. So meaning that placing set amounts of ATP or set amounts of bacteria, either which are swabbed from surfaces or dropped directly over the swab, do not have the same results each time. And also every now and again, randomly high or randomly low results can occur. The last thing is that the devices are not consistently good at quantifying very low RLU numbers, even though they're very good at detecting low amounts of organic matter. So the swab testers are bulk sampling devices. So swabbing a larger area will result in higher RLU readings. In order to compare results, the swab area and the techniques need to be consistent. And then one of the surprising things that I learned was that the relative light units are not a standardized measurement. So this means that results from luminometers can only be compared to identical devices from the same manufacturer, as the ratios used to calculate how much light equals 1 RLU may not be the same. Okay, briefly on swabbing. In wet versus dry swabbing, lab experiments generally show that wet swabs have more uptake on surface contaminants than dry swabs. But in similar tests conducted in conservation, there's less consistent results. Swab efficiency or uptake is also influenced by things like surface texture, swab technique, for example, if it's dragged or rolled over a surface, how much pressure is applied, and then the material or the shape of the swab. Okay, and lastly, in terms of the literature review, I looked into current health guidelines and benchmarks to see what can be considered a good relative light unit target. But in short, there's no current established benchmarks or limits on what is considered a safe or acceptable amount of fungal exposure. So as an example, in 2009, the World Health Organization made guidelines for indoor air quality. And they noted, and this is a paraphrase by the way, that yes, there's strong evidence regarding the hazards posed by biological agents in indoor air, but it's hard to quantify specific microorganisms that are responsible. And this is because people are often exposed to multiple agents simultaneously. It's complex to measure exposure ethically, and there's a large number of symptoms and health outcomes that are a result of exposure. Some organizations do recommend limits for airborne fungi, but they're not definitive or universally accepted. So with this in mind, it does seem quite difficult to make specific benchmarks or specific swab tester benchmarks for conservation use. I don't expect that we can outthink the whole health sector. However, this did eventually lead Crystal and I to thinking about potential alternatives to benchmarks for mold remediation and conservation, which I'll talk about after the experimental bits. So without health-based limits, it's generally accepted by most organizations that preventative measures and minimizing exposure by cleaning mold damaged items is still the most effective solution. And so in a conversation with CCI scientist Tom Strang, we ended up with a very grammatical slogan: lesser is gooder when it comes to mold residues. Looking at remediation guidelines, most will recommend specific levels of PPE, so personal protective equipment, depending on the size of the mold outbreaks. And so I thought this was a nice way to contextualize, since most single artifacts that have mold damage would generally fall under the category of small instance. In remediation or cleaning, most remediation guidance will note that removing as much fungal residue as physically possible is necessary. For non-porous and porous materials with surface contamination only, recommendations generally include drying, then surface cleaning with a HEPA vacuum or other scraping or wiping tools. Treatments such as washing and disinfecting are only recommended occasionally, and then fungicidal treatments are only recommended sometimes, and they're very much not agreed upon. Most remediation documents will also suggest getting rid of porous items with severe mold damage, though this is potentially less useful in conservation contexts. To decide if something has been remediated enough, recent guidelines will generally recommend visual assessment. So the two methods are normally tape lift or wipe test to check if there's any fungal residues left on the surface. Though air sampling exists, it's often only recommended for very large mold outbreaks or where at-risk populations are present. And again, there isn't a clear consensus on what numbers to aim for. Okay, so this background research helped make a framework for the experimental phase. Overall, RLU results are a generally effective marker for the level of organic residues, but they're not absolutely accurate markers of ATP, so the numbers are best used as a relative indicator. The device is also not a measure of fungal counts, but an assessment of cleanliness on a surface. Because there isn't any known safe level of mold contamination, it's not possible to have a particular RLU target which below would be considered adequately mold-free. So to obtain useful results in practice, it's necessary to have a consistent sampling technique using the same area and sampling style or swabbing style. Also, taking multiple samples before treatment and after treatment can help average out the repeatability and outlier issues of the technology. So these key points were taken and directly used to establish the swabbing protocol for the experiments proper. Okay, on to the experiments. I'll briefly go over the swab protocol and then move to each of the separate experiments and the results. So as I mentioned, all the tests were conducted with a Kikomin PD30 luminometer and Lucipac pens, which swab for both ATP and AMP. Clean polyester masks were used to make sure the same sized area was swabbed each time. So you can see that here. The swabs are taken dry. And there's a little diagram here that shows what we ended up calling the CCI swab protocol, which ensures that a sample is swabbed in all directions and would account for things like material grain. Replicates or repeats of the swabs were taken in new locations of the material each time. This is because every time you swab, it cleans the surface a little bit or changes the surface during the testing procedure. And then using what we learned in the literature review, when unexpectedly high or low results appeared, I took additional samples to verify. And then the image at the bottom is just an example of the information that was recorded during the experiment. So the first test probed at the initial question about background RLU levels. So whether we could find consistent background RLUs for new or visually clean book and paper material. Because so many substrates in book and paper conservation are organic materials, sometimes with friable surfaces, we had an expectation that RLU results would probably be different than the background results from manufacturer recommendations. So this was a really simple test. 8 new materials, all stored in the conservation lab storage for undeterminate periods of time. All of them were visually clean without large dust or dirt, and none of them were specifically cleaned before swab testing, which is how we normally use them. I also tested 2 study samples with definitely no mold, but one of them had a friable surface. And each sample had 3 replicates or repeat tests each. So as the graph shows, the RLUs varied between 10 and 750 RLU. And you can see in the black error bars here over each column, the variations between each repeat. Even with this smaller sample size, some of the visually clean materials were edging towards the 500 RLU, and even in the early stages of the experiments, the results were showing us that we may need to break out of the benchmarks set by the device manufacturers. And then, for example, some of the same materials, for example, the oak veneer, which is here, versus the planed oak, show different RLU ranges, even though they're technically the same material. The second of these initial tests looked at if normal lab actions could influence results. So these were taken because normally in book and paper conservation, we touch the objects with bare hands, we speak over them without thinking too much about it. So using the same materials from the previous tests, I did some brief swab tests. For touching, both hands were thoroughly washed with hand soap and dried, and then I spent 15 minutes doing normal lab processes, which is the typical time it takes to regenerate enough finger oils to leave contact traces on paper. And then I rolled all my fingers over the same sample surface. So the results in the bar graph show that yes, touching did tend to cause a very slight increase in detectable organic residues with one outlier result. And the third test was about saliva contamination. The manufacturer cautions in one leaflet against speaking over sample areas to prevent affecting swab results. So to test the extent by which this might happen in practice, I conducted a test over the same rag paper as the previous experiment. So I consumed a set amount of water, had a wait period, placed the sample piece at the workbench, on the workbench at my midsection height, and then standing fully upright, I read one paragraph of text. As with the touch test, there is a slight increase in detectable organic residues overall. So in terms of these three initial experiments, plus the literature review, it showed that using a specific benchmark number might not be as useful as we'd hoped. Also, because conservation actions may slightly alter the results, using a strict benchmark number could be misleading. However, these experiments also showed that taking a swab test from a visually clean location of an artifact substrate and comparing it to a dirty or moldy area might be a better way to get a sense of what RLU might be considered clean or not moldy for that specific artifact. Over the course of the project, I also took the opportunity to conduct quick reference tests of different clean, dirty, moldy surfaces to get a sense of expected RLU ranges with the standard swab protocol. So in one example, swabbing a visually dusty metal shelf had a very comparatively high RLU count to a visually clean shelf. But I was surprised that even in this very large bulk sample, so lots of dust on the swab tip of the dirty sample, it still only showed about 2,000 RLUs. In another example, I took samples from moldy gel and gum and conducted some tests in a biological containment unit. Half of the samples were tested while fresh, and the other half were tested after desiccation or after they were dried. And these results were significantly higher RLUs, with the lowest being 6,000 and the highest being 60,000. I also attempted, because these were so high, I also attempted to take smaller bulk samples by just taking the swab and touching down once with the swab tip, but it still had very high results in the 20 to 30,000s. So based on these results, you can probably generalize and say that fresh or recently desiccated mold will likely have very high RLU results in the high thousands. I also tried using the swab as a diagnostic tool with less success. So as an example of unsuccessful diagnostic results, taking the samples from musty smelling books that required cleaning and airing out resulted in RLUs of less than 1,000. However, a non-moldy but visually dirty book swabbed over 2,000. So in these instances, RLU results weren't definitive enough to help make a treatment decision. There were two instances with semi-successful diagnostic results where the amount of bulk samples suggested a positive or negative result for mold. So for example, in the image on the left bottom, a large amount of powdery white residue would have been expected to result in a higher than 700 RLU if it was mold, just based on the previous experiments. And then on the right, it was the opposite. A very low amount of black residues resulting in 700 RLU suggested potentially it was mold. But beyond these examples, when we were looking at using swabs in a diagnostic manner on actual artifacts, in most cases where the results were in the high thousands of RLUs, which suggested mold, the surfaces already looked mold damage to the trained eye. But I can say anecdotally, it was nice to see a decent correlation. Overall, as a diagnostic device, it's not quite useful unless it's already coupled with existing mold knowledge. And then just a fun fact, the highest number that we've swabbed on this particular device was a dried but very mold damage rolled map, which was about 144,000 RLUs. Okay, onto the case study parts of the experiments. A series of swab samples were taken before and after treatment on paper and wood samples. These were used as initial tests to see if the swab testers could track differences in common mold remediation treatments. So for paper, I used pages from a didactic binding where vegetative growth wasn't visible to the naked eye. There was just some pigmentation on the edges of the paper. And these were chosen for two reasons. The pages with the staining but no visible colonies were slightly more consistently damaged than ones with defining visible mold growth. And then the second was that previous experiments showed that taking large bulk samples of mold would likely have very drastic RLU results. But often when cleaning mold damaged artifacts, mold isn't definitely growing on the surface of each page. There may just be some staining and some small amounts of residues. And in those cases, it's less clear what remediation treatments are necessary. So with the aid of Tom Strang for help with the imaging and Kate Helwig, another CCI scientist who gave us a tutorial on using the large fancy microscope, we were able to take micrograph images to confirm that the pigmented areas of the paper also had fungal presence on the surface. So you can see it in the form of thin branching hyphae and fungal aggregations in the pigmented areas of the paper here. In doing the swab test, each page was tested at 6 locations. So in 2 visually clean areas here, and then in 2 pigmented areas at the right before treatment, and then 2 at the left after treatment. So there were 4 different remediation techniques that were tested, and each treatment had 4 replicates of sample pages, so 96 swabs in total. But for expediency, I'll just note that the treatments were chosen for their relatively common use in book and paper, plus 1 novel treatment, which is the ethanol vapors. So looking at the graph, the yellow bars are the visually clean areas that were tested. The blue bars are the pigmented areas before treatment, and then the green bars are the after treatment tests. So the purpose of the experiment was to see if, in practice, treatment techniques could be tracked with the swab tester, and based on the results, yes, the swab testers were generally able to detect an average decrease of RLUs after treatment procedures. It was interesting to note that the tests with a 10-minute application of 70-30 ethanol poultice showed the most dramatic changes from the before treatment and after treatment samples. And this is presumed to be because both the vacuuming and brushing, as well as the additional application of the 70-30 solution, act as a physical method of fungal removal, even if we discard the disinfecting qualities of 70-30. And then as a quick note, the lower RLU results don't indicate whether the fungi is viable or not, or if it's still present below the surface. It's just what can be swabbed from the top. Okay, and in the case study on wood, which is the final experiment, I've saved this for last because it was conducted in a slightly different manner. This is because the tests were completed at the very beginning of the project before we established a swabbing protocol. And it was done together with furniture conservators and Stephanie Etienne and Crystal Maitland as well. So these samples came from the back of wooden keys from a historic piano, which had some mold damage, and you can see it in the image at the left of the slide. So in these experimental samples, the swabs were taken before treatment, after cleaning with a brush and vacuum, and then after 10 minutes of a type of remediation treatment. And each test was carried out in the same location, meaning that it not only had a treatment, but extra cleaning help from the swab each time. So the results were similar to the experiment on paper. The treatment showed an average reduction in RLU results. However, the wood study showed much more drastic changes before treatment and after treatment. And this is presumably because the initial swab sample had a relatively large amount of vegetative mold on the surface, which was then removed by the swab test itself, then additionally removed after vacuuming and then through the treatments. And the wood surface was also very intact, meaning it was possible to do a very thorough physical removal of fungal fragments and spores without damaging it. So after finishing the experiments and the literature review, it seems that the swab testers are a relatively effective device at tracking mold remediation and cleaning. It can help confirm or reinforce if you've had, if your remediation treatment has had an effect. And I think this is especially helpful in situations where there's a lot of staining and there hasn't been a huge visual change in the artifact after cleaning. I also found when I was doing a collection remediation project after having done these experiments that it would have been nice to have had it on hand to periodically check in and make sure that the cleaning processes I was doing were actually having an effect. So something to help keep on track when processes become repetitive. Overall, it was much less useful as a mold detection device, generally because when swab results were in the high thousands, it was fairly likely to be moldy already, just based on visual inspection. However, for those who don't come across mold as often, it may again just be something that can help confirm or reinforce in particular situations. So just some key points from the research is that a consistent swab protocol is necessary. RLU benchmarks are more of a general indicator than a hard number. And using comparative swabs of visually clean areas of an artifact will give some relative RLU targets to aim for after treatment. I wanted to note for those who are interested in mold detection devices that there is a somewhat similar bioluminescent device that's been recently placed on the market called a mycometer surface. And it works by detecting an enzyme characteristic to fungi. And it works in a similar manner as the ATP swab tester, but with a little bit more pipetting. However, the downside is that they're more complex and they're much, much more expensive. And they have the same caveats about surface testing as adenosine testers. But I've added a reference below for those who are interested. And there are also lower tech ways to determine if something is moldy. Simply taking a small sample and using a microscope with either transmitted or reflected light is a great method. If you can see little colony spots or thread-like branching patterns, it's fairly apparent that you're looking at something different than dust or dirt. Okay, so I'm going to move away from the specific research and talk a little bit more about remediation in general. First starting with the idea of thresholds and then following up with some common questions that came up during the project. So in the process of thinking and talking a lot with Crystal about how much should we clean the surface of an artifact after mold damage, and as I mentioned earlier, how clean is clean enough when there isn't any specific pass-fail targets that say, yes, this is now safe to use, we came up with a framework for thinking about mold treatment in a way that could help conservators make plans for remediation. It's not something that's specifically different from what most of us are doing anyways, but having this defined framework to support how we think about treatment processes turned out to be really helpful in practice. So I'll discuss them theoretically first, and then achieving them in practice, and then about the challenges in the next few slides. So the first, threshold 1, deals with minimizing the health effects of mold to people. This threshold is always necessary to carry out for mold damaged artifacts. To achieve this threshold, the potential spread of mold is significantly reduced or eliminated, and the artifact is stored in a stable environment or one that's not conducive to mold growth. So achieving this threshold means that the amount of spores and hyphal fragments, and therefore any residual volatile organic compounds which can be released from the artifact, are effectively minimized. At this threshold, and until there's more information from the health sector, this may be considered adequately safe for most healthy users, though as a caveat, mold sensitive, immunocompromised, or otherwise at risk people may need to take extra precautions, such as wearing PPE. After threshold 1, there's potential to carry out remediation to a threshold 2 level, which deals with the effects of mold on the collection itself. So this threshold may be something to aim for, but it's not always possible. So in order to achieve this additional threshold, the viability of the fungal fragments and spores need to be eliminated, so killed, so that even in ideal growth conditions, it's not possible for that same fungi to grow. As with threshold 1, achieving this additional level can be considered adequately safe, though with the same caveats as before. Okay, so how are these achieved in practice? In practice, threshold 1 is achieved in 3 parts. So the mold is dried and it's no longer actively growing. It also has to either be surface cleaned to a sufficient level that there's minimal mold residues remaining on the surface, so the surface feels clean and intact to the touch, or one of the methods such as the wipe test or tape lift, or potentially an RLU swab tester shows minimal surface residues. Or it can be cleaned and then encapsulated or separated in some way if it's not possible to do a thorough enough cleaning because the surface is fragile. Then the remediated material is stored in an environment that isn't conducive to mold growth, whatever strategies may work for your particular collection. A practical example would be the piano keys from the case study earlier, where the majority of the mold was cleaned from the surface of the wood just with a vacuum and brush cleaning. In this instance, further measures aren't necessary as it falls in line with most remediation guidelines for mold in non-heritage buildings. Just as a note, this threshold is not fungicidal, but the risk of the same mold regrowing at this stage is minimized by using multiple preventative methods, so drying, cleaning, and ongoing environmental controls. Achieving threshold 2 is carried out by combining the cleaning from threshold 1 with a fungicidal remediation treatment such as gamma rays, ethylene oxide, application of enough heat or cold to cause lethality, or 70-30 water ethanol treatments with adequate contact time. And I'll talk a little bit about more about those last two at the very end. It may be more appropriate to aim for this threshold, particularly in cases where artifacts are severely mold damaged in large-scale mold outbreaks, or for items which will be stored in less than ideal conditions. And I want to note that some treatments such as 70-30 ethanol water solutions straddle the line between threshold 1 and 2, because even though the application of the solution is mainly meant to be a method to reduce the viability of mold, the process also additionally has a cleaning effect, because it may clean or wash the artifact of residues that may not be picked up by standard surface cleaning techniques. So these thresholds are meant to be a bar to aim for, but there may still be difficulties in achieving them. So for example, challenges with threshold 1 are that the symptoms, the severity of the health effects of mold on people may vary widely by individuals. So it's really hard to say when something can be considered adequately safe for everyone. The other difficulty is that porous artifacts are not totally cleanable, so there'll always be some remaining mold fragments, which may be a risk, with the image on the right being an example. Challenges to threshold 2 are that degraded substrates may be more susceptible to mold growth than other areas because it's already been partially damaged and digested. As well, the substrates will also still be susceptible to new mold growth at any time. So the other more important challenge is that sometimes fungistatic or fungicidal treatments are difficult or not possible to apply without further damaging an artifact. So as an example, a document with certain inks may feather from a solvent-based fungicidal treatment, which may be considered an unacceptable loss. So in this case, then going back to threshold 1 and to clean and contain is a potential resolution. So in these cases, with challenges to threshold 2, it's something of what Crystal likes to call a mythical unicorn. So something to aim for rather than an impossible necessity. And then I just wanted to note that there are cases where there are health hazards beyond fungi that may exist. So for example, if there's a flood and your collections are contaminated with contaminated water, in these scenarios, the thresholds may need to be modified to ensure the safety of the users. Okay, this is the last bit. So in speaking with other conservators, we seem to keep coming around to a few common questions and myths about mold. So because I came across a lot of references to key texts while I was doing the research, I thought it would be nice to briefly address them here. So one of the most common questions that came up was about freezing mold damaged artifacts. Does it stop or kill mold? So freezing or low temperature storage will significantly slow or stop active mold growth, and it's a common and accepted method for maintaining stasis of moldy or water damaged items until they can be dried and cleaned. It can be lethal to some parts of the mold, particularly fungal hyphae, which are generally susceptible to freezing because they're more hydrated, but spores or other survival structures may be more resilient. Things that will factor in how effectively lethal the freezing is things like species, the fungal structures presence, the lowest temperature reached by the artifact after freezing, and the rate of temperature change. Another frequent conversation was about the effectiveness of 70-30 water and ethanol solutions, which are commonly used as a mold disinfectant in book and paper conservation. So in case there's anyone listening who may be less familiar, the treatment is a solution of 70% ethanol to water, and it's usually brushed, swabbed, immersed, or sprayed on with the idea that it kills the mold. So in terms of the effectiveness, microbiological research shows that ethanol water solutions have fungicidal or sporicidal properties between 50 and 80% ethanol, and it has the most effectiveness between 60 and 70, so thumbs up, we're doing good. The time that the solution is in contact with the artifact will affect lethality. So As an example, in non-sporing bacteria, 70-30 treatments were shown to be most effective around the 10-minute mark. For mold in conservation literature, there isn't a huge amount of information that I came across that looked specifically at contact times for mold lethality, but one of the few is a 27 research project by Chloe Lucas on using 70-30 vapors to treat mold damaged photographs. Her research showed that five common fungal species in collections, a 2-hour exposure to the vapors resulted in full lethality of the 5. However, when using a tech white poultice, 8 minutes of contact time was only lethal to 2 of 4 species. So I wanted to clarify that in the studies where mold disinfectant treatments are tested, when a treatment is only partially lethal, it may still be a useful remediation technique, as some or much of the fungal structures will no longer be viable. And in the studies, the mold that is regrowing are things like the spores or other survival structures where they're growing in ideal conditions in an incubator. And hopefully your collection spaces are not like that. There's other alcohols tested, which may be used as well, such as isopropanol and butanol, which the details of which can be found in the resources below. The 2012 paper by Silvia Sequeira is particularly informative since it has a very thorough list of historic and contemporary mold treatments mentioned to have been written about in conservation literature, and it includes their advantages and disadvantages. This paper also has one of the most surprisingly comedic things I've seen written in a conservation paper, which is that high-frequency electrical currents were once experimented with as a book drying technique, which also had some fungicidal properties. However, in the disadvantages column, there's a little note that says, a risk to the treatment is that the book may set on fire if it's too dirty. Okay, is using a smoke sponge or eraser effective? Based on the RLU results of the swab sampling and using microscopy, a smoke sponge will clear most of the bulk fungal or some of the bulk fungal residues from the surface of the substrate, provided the substrate's intact. However, it definitely doesn't remove anything below the surface. And if you're only using a smoke sponge, there's a high chance of spreading the fragments around in the air as well as on other parts of the artifact. So it's best as a secondary treatment. Or secondary technique, sorry. And there's a great free mini lecture by Fleur Vanderwood at the YouTube link where smoke sponge clean samples were imaged with a scanning electron microscope and showed some definitive presence of mold fragments and spores remaining on paper. And there's very nice pictures in it as well. Okay, do I need to identify my mold? In most situations where the total mold damage is not extensive, it's impractical and not necessary since all the mold will need to be desiccated, cleaned, and potentially treated with a disinfectant anyways. In really large or extensive outbreaks, that will probably involve buildings remediation professionals, and some organizations will just have specific protocol where they do identify. And this is generally to have to do with making sure there's similar species inside and outside your collection, the indoor spaces to outdoor spaces, to make sure nothing weird is growing in your air systems. There's a few recent exceptions in conservation for identification, which include 2015 work on a Da Vinci portrait to see if anything could be done to reduce or prevent further damage from foxing spots, as well a 2018 study in a collection space identified mold to see why they were having problems after an HVAC renovation. Okay, so I thought I'd end with the last few very basic concerns that we often heard that bring us back around to the reasons why we're interested in mold treatments and health benchmarks. So on the health side, how does mold affect health? And this is very simplified, but it affects people mainly in 4 ways. As an allergen, which causes sensitivity through exposure and antibody buildup, and it can be caused by all parts of the mold. As an irritant, through microbial volatile organic compounds, which are caused by chemical reactions when mold secrete enzymes, which releases gases, and they're often at concentrations that are detectable by humans. So that's the moldy smells. Less common is toxins or secondary metabolites, which are created by some mold species when they experience a certain set of conditions and are generally thought to be a stress response. And then the rarest would be infections. So when a viable pathogenic fungal species can be detrimental, which can be detrimental to humans infected individual. But this is generally less of a concern in a collections context, except with high-risk individuals. And so this leads into the next question. Are documents and artifacts with inactive mold dangerous? And yes, mold that is not actively growing or dead mold is still considered a health hazard, so needs to be cleaned to a threshold 1 level. And then finally, on the collection side, is inactive mold on documents a concern for regrowth? And yes, mold that is not actively growing or documents which have been cleaned of previous mold damage may still be at a risk of regrowth, since there may still be viable fragments and spores. All right, so that last question wraps up the presentation. Thank you so much for listening, and I want to say thank you again to Crystal Maitland and Tom Strang for all their wonderful advice during the project, as well as Christine McNair and Kate Helwig.
Thank you, Tiff. Thank you so much. Oh, wow, there's so much in there. I need to definitely go back and watch the recording and engage with it more. You covered a huge amount of material there. Right, okay, so we'll have a quick question and answer session then. There's a few questions in here already. A couple of people have touched on whether or not the specific species of mold affects the RLU result. And I wonder if you could talk a little bit about that, if that's something that you had looked into or considered.
Of course. So technically, yes, or theoretically, yes, they would affect the amount of RLUs that they show up. So in non-rapid swab tests, which have been done in conservation, they have tested the differing levels of ATP from various species, and they are different. They'll also be different for bacteria and all sorts of other things, which is one of the reasons I feel like using the RLUs as just a general benchmark rather than like a specific number is really important, because we can't really say or identify every single mold that we'll find.
Yeah.
If you're interested in reading more about that ATP testing, maybe email me. I was going to try and say the name, but there's about 4 syllables in this name for the paper that I want to say, and I don't think I could even like make anything come from there.
You must have produced a really extensive bibliography. I wonder if you'd be happy to share that somewhere?
Actually, it is something that I'm hoping to eventually share, is because I did get about 60 to 70 papers that I annotated as a bibliography. So yeah, if you're interested, email me and I'll send you a draft version as well.
Great, wonderful. Thank you. We've got a question here from Bronwyn Glover, which says, you mentioned that the size of the area swabbed will affect the resulting number. How did you determine the size of the area to mask and test?
So the size recommended by the manufacturer is about 10 centimeters square, so 10 by 10. Though when I talked to the Luminultra, the supplier that sent CCI the device, they said that most people swab about a 5 by 5. In the end, we just chose a smaller number that seemed like you could take multiple swab samples from book and papers things, which tend to be smaller. So we just picked a little bit of a smaller one.
You've got to put it down somewhere, haven't you?
Yeah.
Great. Victoria Haddock has asked, do you have any knowledge and or opinions on radiation treatments on moldy paper?
I think that there's really I don't know how to say it. There's quite interventive treatments that can happen for fungicides. So things like what we used to use, ethylene oxide, or radiation treatments. And I think there's a time and place for them. So for example, if you have a really large collection that would be, that had a mold outbreak and it was really hard to freeze or dry them all at once, like that might be an option that you think about. So I don't think any treatment in particular is very bad. It just depends on the circumstance.
Yeah. Okay, great. I've got a question, which is, how did it compare using the ATP testing to using the microscopy as a form of sort of identifying mold, in terms of how easy did you find to use them as to compare them as? Sorry, I'm not very good today.
I think they're both comparable because they both require a little bit of knowledge beforehand and having the device as well. I think definitely microscopy was more interesting to look at because you got to see the molds under magnification.
Yeah.
But yeah, I would say that they're a similar level. If you've never used a microscope before, you don't have a good one available, then RLU swabbing is a fairly simple thing to learn. You probably learn it in about 10 minutes or so.
Yeah, but you felt like you could confidently identify mold using the microscope as well.
Yeah, fairly well enough, and I'm not like super experienced with it either, but after a few goes, it did seem pretty obvious when it was on the surface or just under the surface.
Yeah, that's good to know.
The other thing that I did try doing was using lactophenol staining to see, it's a destructive technique, but it stains the proteins in the fungi, so you can see it very well underneath the microscope as well.
Yeah, oh great, thanks. We've got an anonymous attendee here has written quite a long comment and several questions, so I think I'm going to try and maybe break it down a little bit. On the paper cleaning tests, most of the before and after cleaning results are within error, except the combined vacuum and solvent treatment. Can you comment on this?
Sorry, so in the paper cleaning test, most of the before and after results are within error, except for with the
combined vacuum and solvent treatment, yeah.
I think it has to do with the fact that aside from with the solvent treatment, the amount of residues that was being picked up, like there wasn't enough difference before and after treatment with the amount of residues that were picked up off the surface.
Yeah.
Even in the vapor treatment, because there'd be no cleaning that happened to it, there wasn't a big difference. So yeah, it wasn't a huge before and after. And one of the things that came out from this test was that I did realize that even though I'd kind of thought, oh, this disinfectant treatment with 70-30 isn't that useful in a lot of situations because it's not killing all the mold, actually having something to just help wash away some residues that may be on the surface or physically do something to remove residues was actually very helpful in terms of a remediation process.
Yeah, okay, great. There's a couple of other questions in this question, but I'm going to just skip to another person and maybe we'll come back to them if we have time, just so that everyone can have a sort of fair chance at getting their question answered. We've got a question, let's have a look. Lisa Hanke has asked, would you say testing ATP and or ATP AMP testing can help to identify active or inactive mold?
Like the, I wonder if she means the difference between the two.
Yeah, I think so.
I mean, when I tested historic mold, where I didn't know how long it had been on a surface, there generally was a lower RLU than when I tested mold that had just recently been dried. So I think there would be a difference, but I would say that not having done like hundreds and hundreds of tests, I wouldn't be confident in saying like giving any range at all or saying definitively, but just probably.
Yeah. Okay, great. I've got a question here from an anonymous attendee. What has been your largest mold remediation project and how did you implement a process flow?
So I guess I've had different levels of mold remediation projects. Some where I'm jumping into a process that's already existed. So those are the biggest ones. In terms of when you're the conservator that finds some mold and then you have to deal with it in a large amount of the collection. I actually have a few slides here just in case that this came up. Was basically you want to have a 4-step process and it sort of falls in line with the threshold. So, or well, I guess it's 3 steps if it's the mold's already there. So prevent, stop the active mold growth and remove surface mold and then potentially kill any remaining mold. I'll just quickly go through a few more slides in case anyone else is interested, but I wanted to mention these are all Crystal slides and I've taken them from her, so I didn't make these ones.
Okay.
So the first thing you want to do is, oh, I wonder if this question's more about process rather than the actual steps to take, isn't it? Well, I suppose it can happen in a big, Okay, I'm going to go through these anyway. So isolate. If the mold is active, dry the materials. If you can't dry, then use a system like freezing just to buy yourself some more time. Oops. Then physical cleaning will happen afterwards. and then further disinfection as necessary or as possible. Sorry, if the question asker emails me, maybe I can be a little bit more specific when I understand.
Yeah, sure. No, but that's really useful to see the steps broken down like that. Got a question here from Natalie Brown, which says, "Hi Tiff, thank you for the talk. Have you looked at Rakonarani's study of ATP from CRCC in Paris? They seem to point to the need of deep extraction methods rather than using commercial kits. They have published a study about Petri films and a commercially available alternative. Have you looked into this method?"
I haven't looked into the method specifically. I have read all of her published papers. So I think she's been publishing since 2003 about using different ATP methods. But since I was specifically looking at the use of rapid swab testers, so the ones that just test on the surface, I don't really know too much about the processes or recommendations that she may be using, but I know that they're different. And I think potentially some of them are destructive techniques, and they're more, if I remember correctly, some of them are about using ATP to determine if the mold is viable or not viable after treatment procedures. But I'd be interested to learn more. So if you know of any other papers, please do send them to me.
Great, thanks Tiff. I think maybe we'll just do a couple more questions because we're coming to the end now. We've got a question here from Lindsay Williams, which says, has this been reviewed to a similar degree on other archival materials such as audio-visual films or textiles, to your knowledge?
To my knowledge, it hasn't been. There may well be some research that's been unpublished or other people looking into it. The main conservation study that I know, or two conservation studies that I know that are working with the ATP swabs in a collection, looked at using them for like heritage sites for one. And then the second is in archival collection, but also collection spaces. And both of those come from the Tokyo Center for Research and Conservation. I'm sorry, that's not actually the name of the center, but something like that. And they published abstracts in English and then some papers in Japanese.
Okay, great, thank you. And then if we just have one more question. Abigail Bainbridge has asked, do you have any idea on how the risk of mold regrowth after cleaning and so on compares to the risk that any paper grows mold?
This isn't something I've studied extensively, but just looking at the research where they use different treatment techniques, if the paper has already had mold growth, it does tend to come back much quicker than with a paper that doesn't have any former mold growth. And I suspect it's just because there's so many mold spores in there already, and the paper is already partially digested, so the nutrients are a little bit more available. But again, that's just my supposition. I don't have any specific evidence saying that. Okay,
great. Thanks, Tiff. Well then, I'm sorry, there are still some questions here, but we've run out of time. So if you guys still have questions that haven't been answered, Tiffany's email is up on the slide. So you can have a couple of minutes to copy it down while I just tell you about what the next talk is. The next talk is going to be tomorrow, which is Wednesday, the 27th of May at 4 p.m. local London time, and it will be Josep Grau Beauvais. I'm really sorry about the pronunciation if I've completely butchered that, who is a lecturer at UCL giving a talk on using online and open apps for preventative conservation. So do tune in for that one, it should be really good. So then it just leaves me to say thank you so much to Tiff for your talk. It was super interesting.
Thank you so much for hosting.
So much information there. And thank you to everyone for joining us, and I'll end the meeting there. Thanks guys.